Good evening everyone and welcome to this special webinar from the University of Cambridge. Tonight we're going to be exploring the next frontier in children's health, transforming the diagnosis and treatment of childhood genetic disease. I'm Professor Julian Smith. I have the privilege of being Director of the Cambridge Institute for Medical Research, which is part of the University of Cambridge's School of Clinical Medicine. At CAMR, it's our mission to understand the molecular mechanisms of disease in order to improve human health. And we focus on how cells function and how that cellular function goes wrong in genetic disease and infectious disease. Tonight I'll be hosting the panel with my esteemed colleagues, and just to give you a brief introduction to how the evening will run, I'll do some quick introductions, tell you how the evening will work. I'll then introduce you to my amazing colleagues. They'll introduce themselves briefly, and then I'll get the evening started with some questions just so that you get a feel of the topic and what's going on at Cambridge. We'll then open the floor to you, the audience, to put in questions, and please feel free to do so at any time. So at the bottom of your screen you should see a Q&A box, and you can click on that box and enter a question at any time for any of the participants. That will appear on my screen, and we'll get through as many of the questions as we can in the second half of the talk. Don't worry if your questions, there doesn't seem to be anyone else asking questions. That's how the webinar is set up, so what you're writing is not shared with all of the people on the chat. I should have also said this webinar is being recorded, but again, don't worry, the way the webinar is set up, you can see us, but we can't see you, so your images and what you're doing, whether you're drinking or eating or sneezing, is not being recorded and will not be shared with other people. So we want you to be completely involved and be part of this evening, so please ask questions at any time, and we'll do our best, as I say, to get through them as many as we can. And really, I hope you enjoy the next hour. No question is too big or too small. As I tell my students all of the time, there is no such thing as a stupid question. There really isn't. That's how science changes, by asking questions, and tonight you get the opportunity to take part in that. So tonight our topic is the theme of genetic disease in children. And as humans, our journey through life is shaped by interactions between our genes and environment. And don't worry, we get into more detail about what genes are later on in the discussion. But we're all different from each other, and other than identical twins, all of our genes are slightly different. And some of those variants increase the risk that we might develop certain conditions often later in life. So for example, cancer or heart disease or obesity. And the impact of those variants is often quite mild and quite subtle. But some of us are born with very specific genetic variants that manifest themselves much earlier in life, and they can be much more severe in their impact. And at an individual level, those variants can be quite rare, but across the whole population, there are actually a reasonable number of people who are born with these rare conditions. And these conditions often affect children and often, of course, have large impact on their wider families. And it's these childhood genetic diseases that we'll be talking about tonight, how we diagnose them, how we understand how they work, and how we can come up with new treatments for them. Understanding the causes and the consequences of genetic disease is one of the university's real aims and focuses. And it's also an incredibly exciting and fascinating time for this research field, because technological leaps, which we'll talk about tonight, are having an amazing impact on the rate at which we can diagnose disease, but also how we can understand how those diseases develop, and also how we can develop new treatments. The university's work in this area spans many institutes and departments, including my own, the Cambridge Institute for Medical Research, the Department of Pediatrics, the Stem Cell Institute, the Wellcome MRC, Cambridge Stem Cell Institute to be specific, the Department of Medical Genetics, and many other researchers across the university. Childhood genetic disease is also a really key theme for the Cambridge Children's Hospital, and that's a project which is a very exciting new initiative that you'll hear more about later, and it will really draw on the excellence of both the healthcare and research environment that Cambridge is situated in and add several new exciting strands to it. As all of you know, Cambridge is really a hotbed of innovation and discovery, and you could say innovation is in our DNA. It was here that, using data generated by the amazing Rosalind Franklin, Watson and Crick solved the structure of DNA. It was also here where Frederick Sanger invented a technology to sequence the order of bases in DNA. And years later, that technology was then used by another Cambridge researcher, John Sulston, in partnership with thousands of scientists all over the world to sequence the first ever human genome. And it was also at Cambridge where chemists came up with a completely revolutionary new approach to sequence DNA. And it's that technology that's really driving the revolution in DNA and genetics and genome sequencing, which is changing the way that we diagnose childhood disease and a lot of other diseases as well. So those are the foundations that were built here at Cambridge, and it's those foundations that are leading directly to the kind of revolutions in genomic medicine that we'll be talking about tonight. So that's quite enough introduction from me. It's now my pleasure and honour to introduce my friends and colleagues on the panel who can tell you more about the specific subjects. So just to start things off, I'll ask each of them to just introduce themselves very briefly and what they do. So Professor David Larue, we start with you.
Oh, thanks, Julian. Thanks. Welcome to all the alumni on the call. I myself was a Clare College student in 1984. I did my PhD in Department of Biochemistry. I'm an MD, PhD, and pediatrician, and was lured back to Cambridge in 2015 with the kinds of ideas that Julian just told you about. What's the promise of the new technologies in DNA diagnosis and new therapies for children? And that was such an exciting prospect for me that I took the job as head of pediatrics in 2016. I have a laboratory here in the Wellcome MRC, Cambridge Stem Cell Institute, where I research into the basic building blocks of the brain. And we're looking at now how the interaction of genetics that can sometimes go wrong in children and development can influence our understanding of genetic disease and also come up with potential new therapeutic approaches.
Fantastic. Thanks, David. Next, Dr. Delphine Larue. Dr. Larue is a researcher in my institute at the CIMR. Tell us a little bit about yourself and what you work on, Delphine.
Thanks, Julian. Good evening, everyone, and thanks for joining us. So I'm Delphine Larue. I'm a cell biologist and a researcher at the CIMR, and my lab is studying the mechanisms behind a rare premature aging syndrome called Progeria. So these Progeria are incurable diseases in which children begin to age very rapidly from the age of 2. And what's quite fascinating is that this premature aging, rapid aging process actually recapitulates most of the features that we observe in normal aging as well. And as a result, most Progeria patients die prematurely around the age of 14. So in my lab, by modeling these Progeria syndromes, we are trying to understand how the disease works at the level of individual cells and individual proteins. And this helps us to identify new treatments to help these patients, but that could also have broader applications for aging.
Fantastic. Thank you, Delphine, and we'll hear more about Delphine's work later. And then finally, Professor Evan Reid. Evan, tell us about yourself and your work.
Yeah, so thanks very much, Julian. So I'm a professor of neurogenetics at the university, and my work encompasses both clinical work, where I see patients who've got genetic conditions, in particular neurogenetic conditions, and also laboratory work based in Cambridge Institute for Medical Research. And there, what I do is run a team who study the causes of inherited forms of motor neuron disease, a particular subtype of motor neuron disease called hereditary spastic paraplegias. And we try to look at the internal mechanisms in cells that are causing those conditions, with the idea of understanding those so that we can develop rational therapies for the conditions.
Thanks, Evan. And I think that theme of how the Cambridge environment and how the Children's Hospital really bring together fundamental research, clinical research, to chart the whole story from diagnosis to mechanism to treatment is something that we'll go into throughout the night. So perhaps to kind of get the discussion going, Evan, I'll start with you. can you explain some of the basic science behind both your research and your clinical role as a geneticist? And also, as you do so, perhaps for the people who are listening who know less about DNA and genes, explain what exactly is a gene and why is variation in genes so significant sometimes?
Yeah. So if we start off with the very basics. So the webinar tonight is to discuss genomes. So I guess the question is, what is a genome? So the genome is really our entire collection of genetic material, our entire collection of DNA. And as probably most people know, or many people know, DNA is essentially a 4-letter code. And the totality of that 4-letter code you can think of as providing a recipe by which your body forms. And within the cells of the body, each cell in the body, we probably have something like 37 trillion cells. Pretty much every cell has got a nucleus, and the nucleus is what contains the DNA and the genetic material in the genome. Altogether, if you took the DNA from a single cell and stretched it out, it would probably be about the length of a tennis court. So you can think of the DNA as being a long string within the nucleus of the cells. And within that string, you can think of the genes as being beads within a string, within the string. So when I started in genetics, which was probably about 1995, we used to think we had about 100,000 genes altogether. But with the large-scale DNA sequencing projects that you mentioned before, for example, at the Sanger Centre, we now know that we have roughly about 20,000 genes, and it's that totality of those 20,000 genes which is providing the recipe by which our body forms. And I guess a key thing to say about our genes is that all of them come in pairs, which is in a way a kind of fail-safe mechanism that nature's designed, so that if you have one abnormal gene out of the pair of genes, you tend to be okay. The normal gene can compensate. But sometimes we'll see genetic conditions where one abnormal gene out of the pair of genes is not enough to compensate, and you get genetic disease, or other genetic conditions where you have to have a double dose of the abnormal genes. And what I guess my background has been in gene finding, so to find genes that are abnormal in particular conditions. And that's a very powerful technique for understanding the causation of diseases, because what genes do is they encode for a protein within the cells of the body. Traditionally, particularly for neurological diseases, it's been quite difficult to figure out mechanisms of diseases. But if you know the gene that encodes a protein, then you can find out a lot of information about the protein and what it's doing in the cell, and you can study it really intensively to figure out the mechanisms by which the protein works normally, and also the mechanisms by which it's disrupted in disease. So it gives us a really powerful approach to try and understand mechanisms of disease. And that's really the approach that we take in my lab, to try to get to the bottom, to understand the really deep mechanisms of what's going wrong in the conditions that I study.
Fantastic. Thanks, Evan. I think that central concept that variation between people, and if we look around the world, we see people are so different from each other, and the conditions and diseases they get seem so different. A lot of this is encoded in just variation in this simple code. It can seem hard to wrap your head around. An analogy I sometimes like is that you can think of the English language as a code. It's just 26 letters and some punctuation, but just based on how you rearrange those letters, you can get the works of Shakespeare, or you can get the script for Derry Girls, or you can get the Bible. It's just a rearrangement of the same letters, and it's the same with DNA. It's a rearrangement of the same letters that gives us the differences between
and
us.
the important thing to say is in genetic conditions is often what is introduced you can think of as a spelling mistake, and you can think of examples in a sentence where changing a single letter in a sentence completely changes the meaning of the sentence. So it's those subtle changes can make really dramatic differences in the function of the gene.
So that's the perfect link to my next question. You have been working in this field for a while. How has the technology and approach to identifying these spelling mistakes, these changes, how has that changed in the last 10 years, and what impact has that had on your work?
Yeah, well, so I guess if I go back even further than that, I mentioned I started in genetics up in Glasgow. You probably detect from my accent, not from these parts, in 1995. And back then, we only knew a small handful of disease genes. and the testing for each individual gene was very laborious. For example, if we saw a patient with a particular condition, we would organise a test for that gene. There might not be an NHS service for it. We'd tend to have to find a research lab who are studying that gene, send off DNA from the patients, and essentially we'd then wait probably a couple of years to get results back, and we may never get results back on that particular testing. Compare that to what we can now do. I mean, I have to say, thinking of my younger self back in 1995 and thinking what we can do now, it would be absolute science fiction. I would never have believed that we would be in the situation we are now, which is essentially what we can do is sequence thousands of genes at a time. And we can do that against the background of maybe not all, but we're probably getting close to the majority of disease genes having been identified and understood. We now understand the correlation between abnormality in many specific genes and how they can directly cause disease. So now we can test for those genes using the next generation sequencing technologies that you mentioned before that were identified by Cambridge chemists to sequence thousands of genes at one time, or in fact the whole genome, so the entire 3 billion letters of DNA. And that gives us enormous power diagnostically. So that, for example, if you look at children being admitted to NICU, PICU, the diagnostic rates that we get from genetic testing there can approach 20 or 30% of children in terms of making a diagnosis.
So, I mean, I think that's a really important point to emphasise. I love the way you said your younger self would have thought it's science fiction. I totally agree. The change is just absolutely staggering. I mean, I mentioned earlier the Human Genome Project. It took 10 years and thousands of people all around the world to sequence one human genome, and that was only 20 years ago that finished. And now we routinely sequence thousands, tens of thousands, hundreds of thousands of human genomes every year. So sorry to interrupt. At this stage, I think it would be useful to see what that diagnostic impact that you just mentioned, the ability to identify variants in children rapidly and when they're very young. So we're going to show a short video clip from the BBC about how that diagnosis, the use of whole genome sequencing to diagnose, is now being used in hospitals all around the country.
This is life at its most vulnerable, made even more perilous when the cause of a baby's sickness is unknown. In some cases, it can take months or even years to get a diagnosis, but that is set to change because of genome sequencing.
What are you trying to do to me?
Millie May and her parents, Claire and Chris, each had their genome, their entire genetic code, sequenced to try to discover why she was having life-threatening seizures. The result showed Millie May has a rare form of epilepsy caused by a single gene error, not inherited from her parents. It led to immediate improvements in her care.
Since we've had the diagnosis, it's been a lot better. We had to change one of her medications due to the fact that the one that she was on was obviously aggravating the type of epilepsy that she has. We saw a big difference as soon as that change was made.
It's priceless. That one test result obviously allowed us to put all the correct people in place and make the best for her.
Sequencing the billions of letters of DNA code that make up a genome used to be hugely expensive. Now it costs less than £1,000. Scientists in Cambridge analysed the genomes of 350 children in intensive care, comparing it to the DNA of their parents. They found 1 in 4 children had a genetic disorder and were able to give the diagnosis in 2 weeks. This study shows conclusively that whole genome sequencing is of real benefit to patients, speeding up diagnosis and helping to find the right treatment. From next year throughout England, the NHS will offer whole genome testing to all babies and children where the cause of their illness is unknown, the first national health service in the world to do so.
So just as we come back from that, just a reminder, you can ask questions at any time using the Q&A button at the bottom of your screen. So don't wait till the discussion part. Please ask questions at any time. So David, maybe I can come to you next. So we've heard from Evan about the journey of diagnosis from single genes and long periods of time to this revolution of whole genome sequencing. How's that impacting the NHS and patients right now?
Well, you saw in that video how the Cambridge history in DNA technology is now being embedded into our pediatric practice. And a study that we started in Cambridge with geneticist Lucy Raymond, and now we've continued, it's now recruited over 500 children and their parents, has really had an impact on the NHS. There's been a rapid uptake understanding when whole genome sequencing, a very complicated technique, 6.4 billion base pairs of DNA need to be analyzed. But yet if the benefit is that we can make an earlier diagnosis for an intensively ill child, the NHS takes notice, and now they've made that a national service. So it is the first national healthcare system in the world, the largest, with 66 million people, to offer whole genome sequencing for children who need it. We're finding this technique is really transforming the practice of pediatrics. A child who comes to neurology clinic, we've observed, has a 46% chance of having a monogenic condition, which can be diagnosed by whole genome sequencing. And that means that our practice can never go backwards. We just have to look ahead. For the family, and you heard from Millie May's parents how that diagnosis changed her care, and that's very much the direction of travel we foresee. That based on a really detailed understanding of the child's condition, we can repurpose drugs to be specific and address their particular condition. We can potentially come up with new types of therapies, gene therapies, that will involve the interaction with our basic science colleagues. And one of the great things about Cambridge is we're not a hospital that's isolated. We're very much nearby the Cambridge Institute for Medical Research, the Wellcome MRC Stem Cell Institute, the Laboratory for Molecular Biology, and there's a real interest amongst our basic science colleagues to get involved in understanding the details of human genetics as played out in these sometimes very serious conditions affecting children. And I think another observation is that we are now moving beyond a kind of a problem called the diagnostic odyssey, where the diagnosis of a child with a genetic condition could take months or years. Sometimes, 6 years, a child might come to my practice with a sort of a question mark. That family has been, troubled by what's going on. They've had to spend multiple clinic visits, lots of uncertainty, and we may have missed therapeutic windows. That should be a thing of the past now, because this tool will mitigate that diagnostic odyssey. So we can really look ahead, and I think that's where Cambridge has an important role to play in sort of thinking towards precision medicine based on now what I'm going to start to call the post-genomic era. We're going to take for granted that a child will have a genome sequence, which 20 years ago we couldn't have imagined. Now we have to think ahead to what we do with that information.
Thanks, David. And that's the perfect segue to Delphine. So, as David said, the speed of diagnosis has increased rapidly, and in some cases, as in the video, that diagnosis can lead to an immediate change in treatment. But sometimes the gene or the variant identified, we don't quite know what it does, or we don't understand the impact, and that's where the fundamental biology and the amazing research science that happens at Cambridge comes in. So maybe with the work that you do, can you just tell us a little about how modeling these rare conditions in cells in a lab can tell us more about how the disease works?
Sure. So, once we know which gene and which variant within this gene cause a disease, we can then model this in cells that we grow in the lab. And this is how we can actually understand and study the direct consequences of these genetic variants on how the cells behave and how they grow, for example. And this is the first step, but really crucial step for gaining understanding into how the disease works and why it might affect, for example, specific tissues or organs in the patients. So, for example, in my lab, we have been using cellular models of Progeria that carry the same variants as the ones in children. And as Professor Reed mentioned earlier, in the case of Progeria, it's basically one single change in the whole DNA code. It's a C to a T, that's it. But then it causes the expression of this abnormal protein, which is very toxic for the cells and which will lead to rapid aging. And so basically by modeling this condition in the lab, we are really able to understand what's going wrong in these patient cells. So as Professor Reed mentioned earlier, in the center of our cells is the nucleus that carries the DNA with the genetic information. And the body relies on these cells to divide. And to divide, they must make an accurate copy of this DNA so that every single cell in our body carries the same DNA and the same genetic information. So what we observed in cells that carry the genetic variants that occur in progeria is loss of the accuracy of this genetic information, because we see a lot of DNA damage accumulation and also changes in the organization of the nucleus itself and of the DNA within the nucleus. And altogether, this leads to a rapid cellular aging. And so based on this understanding, my lab has been able to develop creative approaches to try to identify new ways to improve the disease in patients. And so rather than trying to target directly the cause of the disease, so this abnormal genetic variants, instead we've been trying to target the downstream effects on the cells. So in that case, the accumulation of DNA damage and this abnormal nuclear structure and organization of the DNA. And through this approach, we have been able to identify a new drug target that basically dramatically slowed down aging and completely reversed the rapid cellular aging in progeria. So I think this is one example of how modeling a genetic disease in the lab can actually help to identify a new treatment for patients.
Thanks, Delphine. And that journey from diagnosis to understanding mechanism to treatment, and that it happens all within the Cambridge environment, I think is incredibly, it's exciting to be part of. And the Children's Hospital, which we'll discuss in a minute, will just open up more opportunities. Evan, maybe I can go to you just next. I think Delphine touched on this, how we will be able to use the kind of information that comes from these whole genome sequencing technology of children to perform more what's called precision medicine on these children as they come into the clinic.
Yeah. So the idea behind this is to really target treatments based on the genetic information. So to give, I guess, a kind of background example, maybe more from adult medicine that people might be more familiar with, is if you look at patients who are given a drug for high blood pressure, for most drugs it probably works in about 60 to 70% of people, and in 30% of people it has no effect at all. And traditionally what physicians have done in that situation with the people who don't respond is they just go to the next drug. But one of the ideas, or an important idea behind precision medicine, is that some of that variation, whether people respond to a drug or not, is genetically determined. And so by understanding people's genetics, what you can do is choose the right drug for the right person that's going to work much more frequently. So in other words, you're trying to get your response rate up to 100%. So that's a kind of general example of what precision medicine involves. But in terms of rare diseases in children, there are really similar things that are taken into consideration. So the idea is that we use the precise genetic abnormality to select drugs that are going to work for that particular child. And epilepsies are a good example of that, where there are some abnormalities in some genes where we know that certain drugs will work well, and actually some drugs won't work well at all and really would be best avoided. So I think that genetic information, as well as helping with the diagnostic odyssey that we talked about, can really help with the next stage down in terms of selecting therapeutics, helping therapeutics. And I guess as we start to look forward, there are novel therapies that will be coming down, or just starting to come down the line, that really rely on knowing precise genetic information, the precise genetic cause. And here I'm thinking about things like gene therapies, where we've just had the first gene therapy introduced into the NHS for a condition called spinal muscular atrophy. But before you can select children for that therapy, you need to know what the genetic cause of the disease is, so you know which children can be appropriately benefiting from that particular patient, from that particular drug. You can see that sort of thing becoming much more important as we move forward over the years, as we start to use gene therapies that will replace abnormal genes, but also more sophisticated therapies in the future where we'll actually be editing DNA or thinking about altering DNA to correct genetic mutations within patients. So I think there's very exciting times to come in terms of precision medicine.
Thanks, Evan. And Delphine, maybe I can just come back to you next, because I think Evan's illustrated perfectly how understanding the variants can guide to treatments and novel treatments. In the context of your work, how do you go from understanding basic mechanisms to developing new therapeutics, and how does the Cambridge environment help you do that work?
Sure. So in our case, so in my lab, we've been working on identifying new anti-aging genes, and so to this aim, we've been carrying out a whole genome screening approach. So we used cellular models of Progeria, together with the latest CRISPR-Cas9 or molecular scissors technology, to delete one by one every single human gene in the entire genome. So as Professor Reed mentioned earlier, this represents 20,000 genes. So you can imagine that we didn't do all of this manually, as it would take us years, but we relied on the Cambridge environment with the access to high-throughput automated robots and microscopes that were really required for this kind of work. And so we then looked at the cells by microscopy to see the effects that deletion of these genes had on cellular aging and integrity of the nucleus and of the DNA. And through this unbiased, but as you can imagine, very costly approach, we identified 43 genes that, when deleted, actually reversed the premature aging phenotypes in Progeria cells. So that means that they actually made the Progeria cells much healthier. And so, this work has not only uncovered new and completely unexpected information about the mechanisms behind rapid cellular aging in Progeria, but because these genes can all reverse the rapid cellular aging in Progeria, they are all potential new targets for drugs that can benefit the patients, but also potentially that could be applied for normal aging as well. So that's something that we are still currently exploring in my lab. But I think, as I said before, we couldn't have done this, I think, in many other places around the world than Cambridge, due to access to these latest technologies in terms of microscopy and in terms of robotics, for example. But it's also the access to outstanding PhD students and postdocs that are attracted by the Cambridge environment. And they come from a different range of background with a wide range of expertise. And they are able to carry out this kind of very ambitious and very technically challenging experiments. And I think finally the opportunities for close collaborations between clinicians and academics in Cambridge also means that what we do in the lab have a very direct and very tight connection with the disease itself.
Perfect, thank you. So just before we go to questions from the audience, maybe David, I can just finish this section with asking you about the new Cambridge Children's Hospital, and while you're talking, we'll put up an image of what that hospital looks like. What impact do you think it will have on care for children and young people in the UK and also around the world, and how does it exemplify the themes that we've talked about bringing together research and clinical work and the journey from discovering variants through to understanding mechanisms to treatments?
Yeah, thank you, Julian. Maybe I'll just pick up where Delphine gave us that sort of very encouraging picture of the Cambridge community, and how do we inspire young people, and how, if you're a lab-based researcher, do you understand the impact of your work and the potential, synergies by putting the research into the hospital. And that means that the investigator will actually be encountering patients and may in fact be inspired to research a patient's condition. And that is the type of environment that we sought to encourage when we thought about taking a step for the University of Cambridge into children's health research. So this is a new venture for the university, and we wanted to create the ideal environment for that. And we think that is in the hospital. So we'll have a research institute in the Cambridge Children's Hospital. It'll be one of the distinguishing features. It will encourage and incorporate the brilliance that you can find across departments, not just in the School of Clinical Medicine or even the basic science departments. There are roles for engineering. There's actually a role for the law school. And there's a lot of thinking that can really creatively be applied into children's health. And the institute within the hospital, we think, will be the catalyst for that type of new thinking, training of young people, and importantly, coming up with the new ideas for therapies that we can then put into practice. So that's also where it's really important to be in a hospital, where we have a research mission, where we'd like to be at the leading edge of taking the information about a child's genetic makeup and understanding how that can apply to benefit their sort of health and well-being. Understanding that genetics can affect not only physical health but also mental health, that there are complex interactions between what you're born with, nature and nurture, the environment that you may grow up in with the same genetic condition. And Delphine gave a kind of a very interesting example of how you could have a gene that could cause a very serious disorder like progeria, but you might have another sort of other mutation that potentially makes it a little less severe. And we need to understand these sort of balancing mechanisms that can confer resilience to genetic disease. We understand also that depending on if you're in a stressful environment, that could actually make a genetic disease worse. So all of that needs to be considered. And so as part of our research approach, we're looking both at the sort of nitty-gritty biology, but also taking advantage of the health record, the national records that exist in the UK, the advances in data science that allow us to really link nature and nurture in a more complex way than you really could accomplish in many other countries. So this is research that cannot be done in the United States. This is research that can be done in Cambridge, but not at Boston Children's Hospital. And it really takes advantage of the NHS as a single payer system, the national records that exist that are proxies for outcome, and then the, I think, real interest within the pediatric research community to now really tackle these important questions. These are transformative, cutting edge, whatever word you like to use, for the big question in pediatrics. It really comes down to understanding the impact of nature and nurture and using that to better predict and also prevent disease going forward. So that's the ethos of the hospital. We want to apply that equally between physical and mental health. And so that means the hospital also has to have a characteristic that's unique, and that will be the first purpose-built hospital in the world to fully integrate physical and mental health. That involves having two components of the NHS, an acute pediatrics and a mental health trust, come together in this hospital, but with a even sort of stronger commitment to take care of each child, sort of both body and mind. So imagine a child with an eating disorder will have not just a psychiatrist, but also a gastroenterologist on the team. Kind of makes sense. A child with cancer who might have a vulnerability to anxiety and depression will have a psychiatrist or psychologist on their team, and we'll be anticipating those problems and really helping children to cope better, helping to support families. And that also will be a unique feature of the Cambridge Children's Hospital. So we're very excited about this as a new asset for the Cambridge Biomedical Campus. It will provide a vital service for children locally, but because of our innovative clinical model and this sort of integration of body and mind, if you will, and having a research institute of the University of Cambridge in the hospital, we think this will be an absolutely unique and precious new development for Cambridge, but also we believe we can have much wider impact. So that's an introduction. You're seeing some images, which also show how it's going to attempt to be a green hospital. It has some actually sort of like jungle-like gardens. There'll be rooftop gardens. There'll be quiet spaces because, of course, they're very serious, difficult times for patients and families, and they need places to escape and a bit more calming and meditative. That's been incorporated into the brilliant design that now has planning permission from our local council. And this is a project that I think many people describe as charming. It really takes a very few minutes to get behind the Cambridge Children's Hospital, and you can think of it from all sorts of different, unique features that it has. But it really is attracting some very exciting support. We're very grateful to alumni for the support and interest they've shown to the Cambridge Children's Hospital, and I look forward to meeting with people on this call and your friends to tell them about how they could get involved.
Fantastic. Perfect segue, David, to the alumni, the people on the call. So thank you for your questions. Keep them coming in. We'll get through as many as we can. So the first is a very excellent question about the links between childhood disease and adult disease. So the question is, the example about epilepsy is presumably no different in adult epileptics, and can genetics be important for adult treatments as well, even if the variant was originally discovered and explored in children? I don't know, Evan, if you maybe want to start.
I'll have a stab at that. We'll maybe pass on to David as well, who's more of an epilepsy expert, I think. But so absolutely. What we tend to see in conditions that have got a genetic basis is that, on the whole, if you have a strong genetic cause for a condition, they tend to show up earlier and a bit more severely. But that's not always the case, and so there are definitely genetic epilepsy conditions that, while they more commonly show up in childhood, can also show up later on in life. And so, for example, you can see families who have the same gene abnormality in multiple people in the family, where you get quite a lot of variability in the family. So you might have a child affected with a kind of severe onset infantile epilepsy, but somebody else in the family who's maybe only had a much more classical adult onset epilepsy. So so the genetic diagnosis in children and families or in children can have wider implications for the whole family, and can explain other conditions that have maybe been accepted in the family because they haven't been quite so unusual, later onset and more typical of what we think of as epilepsies.
David, do you want to add anything to that?
I think that the question is a really interesting one if you sort of generalize it to understanding the origins of disease. Does that help you better treat, patients or people as they get older? And I think we can start to think about not only conditions like epilepsy, but also, heart disease, maybe even dementia, certainly mental health, as having origins in children and young people, even though the manifestation could be later on. And so I think another charge to pediatrics is, can we develop the tools to understand, the earliest signs or the earliest point when those diseases get started? Would that be a more effective time to intervene? Again, another big question for the type of research that we hope to take on in the Cambridge Children's Hospital.
And Delphine, I think your research encapsulates this incredibly powerfully, where research on this very rare premature aging condition in children probably has a lot of impact, sort of tells us something about how people age as they get older.
So what's interesting is that actually this abnormal toxic protein, which is expressed in Progeria because of this genetic variant, actually is also expressed when we all age to a much lesser extent. But when we look at cells from an old individual with no disease and cells from these Progeria patients, they look very similar in terms of this DNA disorganization and nuclear abnormalities. And in terms of the symptoms of the disease that occur in Progeria patients, they are also very similar to the phenotypes that we see when we age. So, these children develop osteoporosis, cardiovascular defects, which is the main cause of death actually in the patients, loss of hair, loss of fat, and all these kind of phenotypes of aging. So it's definitely a great model to also understand what's going on as we age.
Fantastic. Okay, next question. Clearly you can tell when the Cambridge alumni are on the call, because this is a question about the epigenome. So specifically regarding the precision medicine that we talked about, does the epigenome have to be accounted for? And just for those of you who may not know what the epigenome is The genome is what we inherit at birth, the order of bases in the DNA, but that DNA can be modified in certain ways as we live our lives. Those modifications are called the epigenome. So the question is, does the epigenome have to be accounted for, and could it get in the way of precision medicine treatments? David or Evan, either of you want to take that?
Well, I can take a stab at it. I think, you know, probably the correct answer is we don't know exactly yet, because we're not using it as commonly as we would these genetic tools, or looking at DNA versus the epigenome. But I think pediatric cancer will be a good example for making the case maybe more generally. And we do see that there are certain cancers where if we look for a driver mutation, don't find it, but you do find epigenetic changes that can account for why that cancer has you know, taken off. And there may be more examples like that. So I think we're at the learning stage. It's not a general, you know, go-to test, but Evan probably is better.
I would agree with that as well. So we're starting to introduce into clinical practice epigenetic sequencing panels as well. And they'll, I think, actually allow us to start to address some of those questions. So you could imagine a situation where through your precision medicine, you expect a drug to work, and then maybe we still find in some patients that it doesn't work. Well, maybe then sequencing the epigenome of those patients might start to give us answers, because, you know, perhaps there's some target that drug requires that epigenetically has been silenced in those patients. So I think that's a great question.
And it certainly tells the story of how innovation is always moving. So we've gone through this enormous change where, as David said, genomics is now being integrated into the healthcare service, and we're seeing the incredible impact of that. But there will be another wave after that. And yes, the concept of what we'll be able to do in 20 or 30 years, I think we'll still be thinking it's science fiction, because we can't imagine the progress. Okay, next question. Specifically about epilepsy, in terms of what are the next steps? What do you think, David, are the sort of next big breakthroughs in our understanding of treatment?
Well, it's a very thoughtful question. I think, you know, the low-hanging fruit is now to take the genetic information, look at the drugs we have available, and, you know, use the right drugs and avoid the wrong drugs for each child's condition. Beyond that, we need to look at the sort of the causes of epilepsy, and that's where you do need to put on your basic science hat a little bit and understand the cells that may be affected and the particular reason why that epilepsy has occurred, if it's a genetic epilepsy. And I won't talk about other kinds of epilepsy, but you know, in that scenario, you could have the lack of a good thing or too much of a bad thing. And then we start to think about gene therapies that can either decrease the sort of dosage of a bad protein, or maybe increase the dose of a protein that's needed but lacking. If you start to think in those terms, you can think about many sorts of ways forward in terms of new therapies. But then it starts to get a little bit challenging about how you get the drug that you think can help into the right cells. And classically, you know, pharma has been limited by the blood-brain barrier and getting drugs into the brain. So we need to think about also how can we deliver better to these parts of the brain that may be otherwise inaccessible by typical drug delivery routes. In Cambridge, we are thinking ahead to those questions. There are also companies that are now helping us to think about how we can not only come up with a solution, which may come from the lab, but also, you know, get it to the right cells in the patient where they can do the most good. A technical challenge obviously has to involve both clinicians and sort of some research skills and engineering almost to actually get it where it needs to go. So that's a little bit of a convoluted answer to say that I think we will want to come up with bespoke approaches depending on the type of epilepsy. And there are actually some fantastic options about new therapies that we can think of. They will require research undoubtedly before we can sort of, you know, make the case. Can we start to come up with precision medicine for, you know, the wide variety of different types of epilepsies?
And that again emphasizes this theme that, you know, the new approaches in some cases will lead to new options rapidly, but other cases they'll point very clearly to new research directions, and those research directions often will need to be interdisciplinary, bringing together engineering and chemistry and biology, and that's something that Cambridge just excels at. So
I agree.
Fantastic. Okay, another specific example. So we've talked a bit about epilepsy and neurological diseases, but what about metabolic disorders? What are the potential impacts of this approach for metabolic disorders? And the question is specifically about glycine metabolic disorders, but maybe we can broaden it out.
Yeah. I mean, I guess genetics has made many advances in our understanding of metabolic conditions. So as a clinical geneticist, metabolic conditions are somewhat daunting because there are a huge range of very rare metabolic conditions. But again, so they've traditionally been difficult to diagnose. But the whole genome sequencing technologies actually really massively simplify that, because you've got one test rather than potentially tens or hundreds of tests to allow you to diagnose metabolic conditions. And genetic or identification of disease genes over the past 20 years has identified many or most of the genes that are involved in metabolic conditions. Even if you think about common metabolic conditions like diabetes and so on, we know of quite a lot of genes that are associated with some very specific early onset types of diabetes, and other genes that are associated with conditions like lipodystrophies and so on, which again you can think of as metabolic conditions. Again, some great work in Cambridge identifying those genes by colleagues in the Institute for Metabolic Science. It's located just a few hundred meters away from where we're speaking.
Maybe just to address that specific point about the kind of glycine metabolism, there are many rare disorders. They're very specific, and the world's expert may or may not be in Cambridge. I mean, I've been surprised when I've been asked questions like this, how many experts are in Cambridge, and I didn't know that they were here. And so that's a great pairing, or a great discovery if we can find that person already here. If not, we need to think about international partnerships, and we need to look at global expertise. And I think that also has to be part of our approach. We can't do everything here in Cambridge. We need to think globally. We need to think across populations. And there's a great already sharing of genetic information with clinical minded people who know that this is going to benefit patients. I think that can also bring in the expertise to look at specific metabolic problems like glycemia. But there's also the potential for us to recruit and develop new programs. So I think we're always interested to talk to families and see how our research programs can evolve to address some of those needs.
And I think you've just emphasised something that is really important, that this is, Cambridge is an amazing place to do this work. It brings together an incredible number of strands, but it's an enormously globally collaborative endeavour. And all of us work with outstanding researchers and clinicians all over the world. And that's really what drives the field as well, because it's only by sharing approaches and information that we really come to some fantastic discoveries. So sadly, that's all that we have time for tonight. I'd first of all like to thank the amazing panel, Dr. Larue, Professor Reed, Professor Rowich. Thank you for your time, sharing your insights and expertise. I think we've really seen how the whole genome sequencing and how it leads us to treatments, and also understanding cellular mechanisms, and how within the Cambridge environment we have the opportunity and are bringing those together in an extraordinary way, really is making a difference already in children's and families' lives, and how the Cambridge Children's Hospital has the potential to have even more of an impact. And the little sense you gave of what an extraordinary building and integration of research and healthcare that will be, I think, is incredibly exciting as someone who works here, and I'm sure for the people on this webinar as well. Thanks to everyone at home for joining us for this evening, for contributing questions, for being part of the conversation. We hope that you've learnt something. We hope that you get a snippet of some of the sense of just excitement and innovation and change that we're living here at Cambridge. It's really an extraordinary time to be working in this field. If any of you are interested in finding out more about any area of the work that was covered today, please feel free to get in touch. You'll get an email after the webinar with contact details to get in touch with, and I know my colleagues and friends and others within the environment will be really happy to talk to you and share more about what we do and what we think that the future is. So thank you very much again, and have a lovely evening.